Today we'll discuss vesicular liquid biopsies for pregnancies, which can be used as a non-invasive monitoring system for the quality of pregnancies throughout the pregnancy. I want to start with a review of the clinical and scientific approach to give you an idea as to why we would use this approach in the context of pregnancy. Preterm labor and preeclampsia comprise two of the so-called great obstetric diseases. Though the clinical presentation of preeclampsia is distinct from preterm labor and should not be confused clinically with it, it is believed that these two great diseases may share common pathogenic mechanisms which may arise secondary to aberrant placentation during the first trimester of pregnancy. Cytotrophoblasts normally invade the decidua of the uterine lining to transform the uterine wall and lining during placentation. The arrangements in vascular remodeling and other transformations in the first trimester may present a root cause for both preterm labor and preeclampsia, all set up by the end of the first trimester. Placenta accreta is yet another serious condition for pregnancy, whereby the placenta grows too deeply into the uterine wall. All three conditions may benefit from early, minimally invasive methods to detect pathogenic mechanisms affecting the maternal-fetal interface. This enables early interventions, say by 16 weeks, that could reduce severe outcomes to both the baby and the mother. Nano and microvesicles, also called exosomes or microparticles, are a regulated form of information exchange. They provide another means for our cells and tissues to respond to physiological stimuli or disease. They relay that response locally or at a distance, and they deliver biological payloads to cells and tissues. These little guys are not rare. In fact, they are surprisingly abundant, to the tune of billions or trillions per milliliter by some estimates, and they carry a rich source of biological content, exciting stuff for biologists and diagnosticians. Again, the truly important point for this talk is that they are biologically active, and for this conference group, they harbor a rich source of proteins. Especially interesting is the cellular content, which allows us to assess cell signaling and other such interesting phenomenon. During pregnancy, the placenta and maternal-fetal compartment may contribute at least half the number of circulating exosomes. While sampling the womb and its environs, the exosomal MP content will additionally be sampling systemically, providing information about the mother's current condition and reaction to the pregnancy. Monitoring the pregnancy non-invasively is our goal, and we particularly want to do this early in the pregnancy, in the first trimester. An effective liquid sampling or biopsy avoids needles and villus or placental sampling and the attendant dangers of that. I'll now discuss the basic technology and give an assay overview of how we collect the data and do the analysis that I'll be showing you later in the talk. The platform we are using for particle analysis is based on two steps. For one of them, I am lumping together several substeps, as you'll see. Initially, there's the exosome microparticle isolation step, and then there's a mass spec proteomics analysis step. The overall workflow, as seen here, is really not unlike, conceptually speaking, a typical genetic testing or next-gen sequencing workflow. Start again. In both cases, you have an isolation step of the content to be analyzed, a preparation step for analysis, and an analyzer for that content, and then the informatics software step and an interpretation and a report generated. Specifically for what we do, we use custom-made columns from a GMP manufacturer in Massachusetts for isolation, a patent-pending method for prepping the proteins for analysis. We use a targeted MRM approach to quantify the established markers for preterm birth. An algorithm is used to score the values for risk stratification. The nexosome columns we use for isolation are a modified size exclusion chromatography column with some bead specializations and a proprietary buffer to optimize particle enrichment and yields. About 1/2 milliliter of blood is used in the current configuration. As seen here, we have a red graph with optical measurements under the modified SEC graph. And that tells us where the vesicles are. That is overlaid onto a protein concentration measurement, which can be seen to the right. There are 24 fractions collected. Now that's more than we usually collect for a clinical assay, but we do this for QA. The 2 peaks indicate where the nano microparticles are and where the plasma proteins are. And that is the second peak to the right. That indicates the plasma proteins. The shoulder next to the peak, that second peak, represents the particle protein, which of course overlaps the particle fractions indicated by the red peak. We have on the right proprietary dark field hyperspectral imaging methods to visualize nature's beads and software to size and quantify them. So what you're seeing is a mass of microparticles and nanoparticles, mainly exosomes and mid-sized vesicles spread out over a slide. This came from one of the fractions. That next panel on the right just indicates the software, which can size and count these microvesicles and exosomes so that we can identify and quantify them. The mass spectrometry protein prep step includes patented online cleanup that avoids multiple purification and enrichment steps offline. There is minimal sample loss this way, and we don't have to remove membranes, lipids, nucleic acids, small molecules, and that sort of stuff. We get higher resolution, about 10-fold signal intensification, shorter run times, and other benefits by using our particular setup. And there are patent-pending methods associated with this. The analyzer used is the TSQ Altus MD. And the main point I want to make here is that we use what's called NormaFlow by the Vanquish UHPLC, rather than nanofluidics, which would take a very long time per sample. This gives us run times of 10 minutes or less that are comparable to other run times for clinical assays, such as vitamin D measurements and toxicology. The mega enrichment we get with the columns allows us to do this. And the CVs are very good over the entire process. That is from collection all the way through analysis, as you can see over on the right, generally in about the plus minus 10% range. We worked very closely with Dr. Zhen Zhang from Johns Hopkins. He's an excellent bioinformaticist and biostatistician. We used several AI methods and rigorous statistics to build our classification models. This allowed us to stratify patients as high or low risk, and we now can issue a risk-based score lab report that's indicated on the right. I'll talk more about the details of the data, specifically the second panel to the right, in just a moment. I just want to point out with this slide that we collaborated with Thermo Fisher and its BRIMS on the preeclampsia project I'll talk about later in the talk. We did the particle isolations ourselves and then sent them those isolates. They did the MS discovery work analytically. Tom McElrath, Dr. McElrath from the Brigham, did the informatics and classification work. And we had some input from Dr. Zhang. I now want to move on to a review of the actual clinical data that was derived from those methods. The preterm birth study followed a systematic discovery, qualification, verification, and validation scheme. I'll tell you more about the extended validation we're undergoing in a moment. Validation that you see here, the third paper from 2019, was a multi-site validation, and all those blood samples came from similar collection protocols, and all were from the first trimester, about 10 to 12 weeks. In addition to an all-moms panel, we whittled down from a larger discovery set to a 4-marker panel for the validation study shown here. Those markers are IC1, LCAT, TRFE, and ITIH4. Those markers correlated with known pathways in the pathophysiology for preterm birth, including oxidative stress, adaptive innate immunity, trophoblastic function, and placentation mechanisms. That panel classifies specifically first-time mothers, which are an important unmet clinical need. Because there are no known markers for that subgroup, and I can get to that in a moment. ROC curve to the right indicates the performance of the panel, the new mom panel, and the algorithm performance for new moms versus all moms. The new mom ROC is indicated in red. We have a second panel that's useful for all mothers, regardless of parity, that is, whether the mother had a baby previously. Kaplan-Meier curve to the left illustrates two populations separated out by gestational age at birth using the risk stratification model. The red indicates the low-risk births, and the blue line indicates the high-risk births. The model works pretty well, especially considering that these are asymptomatic patients in the first trimester, with an event that occurs or doesn't many months later. In fact, the performance of our model is quite better than current assays indicated here, including clinical parameters and biophysical approaches that predict or diagnose preterm birth and versus term births. The extended validation we are currently involved in is the largest of its kind, using a government-funded biobank that was almost tailor-made for our panel and validation studies, the so-called New Mom to Be cohort, PI being Dr. David Haas at Indiana University, who is another academic collaborator. Almost 10,000 first-time moms were participants with an extensive data collection and multiple sample types collected at defined intervals throughout the pregnancy. The NIH randomly selected about 2,400 of the participants. The breakdown of normal versus disease or birth complications is shown here. We have broken up the validation into 3 phases, and we have recently completed the data generation for the first and second phases. Data analysis is ongoing. All I can say right now with this slide, which is very preliminary, is that using the same algorithmic model from the 2019 paper, which we call Study E, using that model, we recapitulated the results when looking at the early data from this new cohort. That model stratifies risk for early births, especially very early births in this new cohort. The development path for a homebrew or lab-developed test is shown here. It shows where we've been, documented, and published, who our partners were, both public and private, that are critical to our success, and where we are going to release our first-generation test, which may be within the next 6 to 12 months. Okay, this slide is just a reminder to reintroduce the preeclampsia collaborative study between NX Prenatal, Brigham and Women's, and Thermo Fisher. The preeclampsia study involved David Saraceno and the Brims. We used the same nexosome isolation protocol for particle isolation from a first trimester preeclampsia cohort. I sent those samples, and he fed those into a Vanquish UHPLC coupled to a Fusion Lumos. Dr. McElrath at BWH performed the statistical analyses on about 800 to 900 protein candidates, identified by first creating libraries and then performing the comparative proteomics and using the libraries to confirm IDs. The takeaways from the study are, first of all, a clinically relevant set of markers that can stratify patients for preeclampsia, specifically preeclampsia before 35 weeks. More interestingly, these markers can stratify for more severe clinical presentations. For example, the mean blood pressures are higher in cluster group 2, and that group also has higher 24-hour urine protein values. Both of these are classic definitions of preeclampsia. Cluster 1 was associated with a less severe phenotype and a platelet-centric biological pathway mechanistic structure. Cluster 2 was identified with a complement-centric biological pathway annotation. Both of these clusters make sense clinically, and they both suggest specific therapies. A further demonstration of the generalizability of the approach is the most recent collaboration between NX Prenatal, Brigham and Women's, the Beijing Genome Institute, with support from Thermo Fisher. The placenta accreta study. As I said earlier, this involves a placental encroachment deeper into the uterine smooth muscle wall. The outcomes of such a disease have potentially severe consequences, including massive hemorrhage, unintended hysterectomies, and death. We worked our nexosome isolations into the BGI workflow, which involved both their data-independent and data-dependent acquisition processes. The idea was to create accreta versus normal libraries and use those for IDs during the profiling of the particulate samples. This data just hit us a few weeks back, so I'm not really going to show you classification data, which is ongoing. But what I will show you here is the type of information you can get from these little vesicle buggers and the biological and basic insights possible. First, the hits, the proteins themselves, and the pathways you get are often cellular, very relevant, of a signaling and metabolic pathway nature. They are not few. Many very interesting pathways are seen, such as those in cancer development and invasion and metastasis pathways. And this is not irrelevant for trophoblastic disease of an invasive phenotype. Indeed, this is like getting a cellular and tissue biopsy of protein content, but you get this peripherally. What I'm showing you here is a very interesting example of a signaling circuit diagram that was hit multiple times by different proteins. I have intentionally de-highlighted important hits in the smooth muscle contraction pathway, which is actually relevant to both vascular and uterine smooth muscle contraction. Vascular development and regulation is highly important to both placental development and parturition or birth. I can just tell you that the key signaling regulators include G proteins, kinases, activators of kinases, myofilament regulation, and several new objects of interest. And I can tell you we'll have a lot of fun analyzing that data. So as an overall summary, and to wrap up, I'd like to point out that we have a distinct early warning system that follows a logical progression of monitoring prenatal care. We are sampling important biological processes and the crosstalk between the mother and the child, as well as systemic information and the reactions to the maternal-fetal environment by the mother. A non- or minimally invasive biopsy technique, depending on the sampling of micro and nano-vesicular content, will be very useful for this strategy. Thank you.